Welcome to everybody. Today's webinar, as said, will focus on the role and the advantages of using biological markers in the study of inherited ataxia as a part of the educational program offered by AGI to young investigators. We will take questions at the end of the presentation in a dedicated Q&A section, but you can also use the chat to make questions and comments, and then we will take a note of those. Next slide. I'm Filippo Santorelli, with my colleagues David Mengel in Tübingen and Puneet Opal in Chicago. We present a specific outlook. What are biomarkers and why they are so important and necessary? How to collect informative biological samples, and how good biomarkers are applicable in clinical and research studies. The learning objectives of today's presentations are the following. We will describe categories and context of use for fluid biomarkers. You will learn how importance of uniform standard operating procedures for collection, processing, and storage of biosamples, and also examples of candidate biomarkers for clinical studies. In other words, example of biomarkers used for trial readiness. In the clinical research of inherited ataxia, there are several challenges. And among those, major issues are related to the low disease prevalence, although they are collectively the number of patients are collectively huge. Single forms are individually quite low, with an estimated prevalence between 1 and 6 for major SCAs. There is also an important clinical and genetic heterogeneity, and there is a lack of objective disease measures. That's why both government and private and also non-private funding associations support programs for the development of well-validated preclinical data, both in vitro and in vivo, using animal models of hereditary ataxias. There is also effort to define better clinical measures, such as rating scales or wearable devices. And also, there are efforts for a more specific trial design. And of course, the definition of biomarkers of disease at several levels. In this case, today we will present only information related to fluid biomarkers, but there are also other biomarkers, including imaging biomarkers. Next slide. What are biomarkers? From a, let's say, best definition from the biomarkers endpoints and or other tools, resources, FDA and NIH Working Group, the definition biomarker is defined characteristics that is measured as an indicator of normal biological processes or pathogenic processes or responses to exposure or intervention. So in other words, biomarkers refer to a broad range of measures capturing what is happening in a cell or in an organism at a given specific moment. And is something that is measurable, can be measurable, but is not how it's not the way to define how a person works or feels or functions. Biomarkers are objective medical science and are also used to measure the presence and the progression of disease, but there are different categories. The set of biological markers is important. Next slide. the set of biological markers important in inherited ataxia in other neurogenetic conditions can be split in different subgroups. We have diagnostic biomarkers where diagnostic biomarkers are used to detect or confirm the presence of a disease or a conditions of interest, or to identify individuals with a subtype of a disease. An example of diagnostic biomarker is sweet chloride that maybe is used as a diagnostic biomarker to confirm cystic fibrosis. Another category is monitoring biomarkers. Monitoring biomarker measure repeatedly measure repeatedly for assessing status of a disease of a specific medical condition, or for the evidence of exposure or a medical product or an environmental agent. Monitoring biomarker, example of monitoring biomarker is PSA, prostate-specific antigen, that may be used as a monitoring biomarker when assessing disease status or the burden in patients with prostate cancer. Response biomarker are also used to show that the biological response, potentially beneficial or harmful, has occurred in individual who has been exposed to a drug or to an environmental agent. As example of response biomarker are also surrogate endpoint biomarkers that are used to substitute for a direct measure on how the patient function or responds to a specific drug during a clinical trial. But also pharmacodynamic biomarkers are also responsive biomarker, are considered response biomarkers. Predictive biomarkers are another category that is important to know. Predictive biomarkers are used to identify individuals who are more likely than similar individuals without the biomarker to experience a favorable or unfavorable effect from exposure to a medical product or to an environment agent. Britta, I think you can have the poll question here. Predictive biomarkers is used to identify individuals who are more likely to respond to exposure to a particular medical product or environmental agent. And the response could be a symptomatic benefit or improved survival or even adverse effect. Prognostic biomarkers are used to identify the likelihood of a clinical event or a progression of the disease in patients who have the disease or a medical condition of the interest. So in other words, prognostic biomarker is the one that indicates an increase or decreased likelihood for a future clinical event or progression of the disease in the identified population of your study. Safety biomarkers is one of the more important categories when treating biomarker categories when considering clinical trials, and is biomarker measured before and after an exposure to a medical product or an environmental agent to indicate the likelihood or the presence or the extent of toxicity of the specific drug or a specific adverse effect. Example of safety biomarkers, very common example of safety biomarkers are hepatic aminotransferases that may be used as safety biomarker when evaluating potential hepatotoxicity in patients. Susceptibility and risk biomarker is the latest category that indicates the potential for developing a disease or a medical condition in an individual who has no currently that specific clinical condition or does not have that specific clinical status. Very common risk biomarkers are in the oncogenetics, the variants in the BRCA1 and 2 genes that may be used as a risk biomarker to identify individuals with a predisposition to breast cancer. Then I think David. The vote of the poll has been summarized. So the question was biomarkers are relevant in the following areas, and the question is 100% of the audience responded susceptibility or risk of developing a disease. That's correct. Or 94% to identify a disease prognosis and monitoring, and 88% consider biomarker relevant in the area of disease diagnosis. A less percentage consider biomarker relevant as a biological fluid necessary to determine the safety of a specific drug. Okay, so I think David is your turn.
Yeah, thank you to have the great opportunity to speak to you today and discuss a very important topic before we can analyze any biomarker, and that is how to collect informative biomarkers. And imagine you were to set up a clinical trial, a cohort study that goes year-long, and you also want to encourage people to collect biomarkers and to collect fluids for them. One of the first, you may already have some analytes in your head that you want to analyze. Let's say you are interested in analyzing NFL via SIMOA technology, so you know you need plasma. But there is a lot of other techniques to analyze a broad spectrum of biomarkers, including proteins, metabolites, RNA, and you can use both unbiased and untargeted techniques. And maybe in the future, there's also techniques that you're not aware of at the moment. And some of these techniques might require special biofluids that work well with one method, but not with the other one. And also some methods might require certain processing steps and pre-analytical factors to be considered before you do that. It means before you set up your trial that goes year-long and includes many centers, you have to think about which kind of methods might be possible and consider this in planning. Which kind of biofluids could you collect? You could collect cerebrospinal fluid. It's a very informative biomarker, but not so easy to get in patients, especially not if you want to do that repeatedly. You can collect urine, and you can collect what we always do, you collect blood, and depending on the anticoagulant and depending on the processing, you can then have serum, EDTA plasma, heparin plasma, sidered plasma, or dried blood spots. Because I've even read this sometimes in papers as misconception what serum and plasma is and what differences, I will just explain it for a second. So if you just collect blood into a tube and you let it coagulate, you don't put any anticoagulant in, then you get serum. If you spin that down, you take the upper phase, you are done. You basically have plasma minus clotting factors. If you add anticoagulants, and that can be EDTA, sidered plasma, or heparin, then when you spin that down, you get three phases. You get red blood cells, you get white blood cells, you get plasma, you take the plasma, and you have your plasma samples. Another method that we would like to encourage you to think about is also dried blood spots. The traditional way of taking dried blood spots is just taking the blood on a Whatman filter, let it dry, and later on you can do analysis. But there's also another emerging technique that is Noviplex cards. Here you put your blood spot on the card, and the card actually separates through a filter red blood cells from plasma. And if the plasma is then dried in the card, you can strip off the top foil and you basically have plasma in your cards. So that opens you a lot of opportunities, because in plasma, it has been shown that you can, for example, using these cards, you can analyze NFL levels. That means, for example, if you have a patient population with a genetic disease, and you might want to start treatment at some point where there is an increasing NFL concentrations, for example, nowadays you would have to let these patients come to your specialized center, let's say every year. With these kind of cards, you could potentially put that part to a general practitioner, or even maybe at home use, and then people could send in the cards and you could analyze them and then ask them to come to your clinic when it's necessary. So there's a lot of different options that you want to consider now when studying cohort studies, which should be collected in these studies. Another important aspect that you need to consider is you have to look into fluid matrices. And because fluid matrix, and what we mean by fluid matrix is, do I look into citrate plasma, EDTA plasma, serum heparin? There's actually differences in the levels of biomarkers that you can detect depending on technique. I've put one slide here on the upper left side, which is NFL that I have measured here as an example in control individuals. And as you see is that NFL levels are actually quite different depending on which kind of matrix you use. That means if you, for example, plan a study, let's say in study A, you get cerebral plasma from patients and controls or only patients, and you get serum plasma, a serum from another study, you might have problems combining this just the way it is. Another example I put on here is I did a similar experiment in mice. This is actually scar-free mice. And what you see here is that, again, in mice, you see differences when you use citrate versus other matrices. Upper left panel is a study that I did some years ago, where we looked into, we developed a new assay for a tau fragment that now has major implications in the diagnosis of preclinical Alzheimer's disease. And what we looked into is which is the ideal matrix to use for this assay. And what you can see is that if you use EDTA plasma, you can readily and you can very reliably detect these levels, all of these in these patients with the assay. And the dashed line is actually the lower limit of quantification. If you use serum, you can hardly detect it because with most patients you are below the LOQ of the assay. So using serum would not work well with that assay. Another example is if you're interested in looking at RNA, there's an example here in quantitative real-time PCR looking at microRNA 21, a microRNA 1. You see if you use EDTA, lithium heparin serum, you get the problem that you don't get measurable levels of lithium heparin of both microRNAs. The reason is that heparin actually inhibits polymerases, and that if you just use lithium heparin, you can't detect anything. For sure, you could use heparinases and things to get around that problem, but it would be much easier if you would have collected EDTA or serum right from the start. Apart from considering matrix effects, there's tons of pre-analytical factors that might affect biomarker analysis. Let's look at CSF first. For example, if you want to collect CSF, it makes a difference when you have ever done this. Sometimes you get some blood contamination because you hit a vessel. Actually, getting some blood into CSF can affect your biomarker level depending on what you want to analyze. So it would be important to track that, or to know, or maybe even disregard the example if it's too bad. Plastic can matter depending which tube you use. In Alzheimer's disease, for example, we typically measure like A-beta proteins, and these are sticky. If you use certain plastic material, the protein will stick to the wall, and levels will be lower if you use one certain type of tube. So you want to make sure that you use the right tube in that case, and it's standardized between centers. Needles can matter, the needle size, because depending on the needle size, you may have more or less hemolysis going on, which can affect your levels. Obviously, it matters which kind of processing, how you process the samples, the time, the speed, the temperature, how samples are being spun down before you can take the plasma or serum out. Aliquot sizes matter. Even small things matter there. So when you imagine that you have, depending on what kind of tube you use for freezing things and the volume you use, the surface area of the volume of the CSF, for example, versus plastic changes depending on when you change those both parameters. And the higher your level of surface versus volume is, the more likely things can stick to plastic while freezing it for prolonged times. Storage temperature measures, but I think -80 is kind of standardized now for many reasons, and freeze and thaw cycles numbers matter. Sometimes some analytes they go away, or they suffer if you freeze them for too often. And one way around that is that before you start your study, you can ask the partners who want to do the study with you to do a lot of aliquots right from the start, because then you can give your collaborating partners just one aliquot. They freeze and can do the analysis, which is much better than just having one, which you have to freeze them for all the time. Here's one study where I actually checked one pre-analytical factor in one of my analytes, which is the N-terminal tau assay. And here we took plasma from individuals and we checked what happens if we process them in less than 2 hours, which is optimal, which we can surely, which you can provide if you are a specialized center for this. Next condition, 6 hours, so it takes a little bit longer. The plasma sample is lying around, and then the study nurse takes some time to bring it to the department and process it. And that is 18 hours. In 18 hours, we chose, that means like you collect your plasma sample, you put it into a fridge, and then you process it next day. And as you see here, there's actually 5 individuals. We checked tau levels in these individuals in these conditions. And what you see here is that in 2 individuals, we get substantial variation of actually 40% when you have prolonged times before you can actually process your samples. And importantly, what you see here, there is actually an individual difference, and that's a huge problem. Just imagine you would analyze the last time point from all of them, and across different patients. That's a problem, because the effect depends obviously on other factors that you can't take into account here. So what's the consequences for you, and that we are considering in AGI, and that we want to tackle, and that's why we meet, and that's why we bring up SOPs to make sure that we don't encounter these problems. We have seen that fluid matrix has an impact on biomarker levels. On the other hand, we have seen that some techniques require a certain fluid matrix. How can we tackle this? One way is that we sample a maximal broad range of material. So you sample CSF, you encourage people to collect serum, different plasma matrices and urine from patients and controls. So then you have all your matrix in place that you need once in the future a very interesting assay arises. We have seen that pre-analytical factors influence biomarker analysis. So how can we tackle that? I mean, the way to tackle this is that we well harmonize sampling, processing, and storage between centers. We have taken that approach, and we have started to discuss this in the AGI Working Group, and have made some suggestions already, where this is just a glimpse for you to see that this exists, and we're working on this, that we kind of suggest which materials should be collected. We suggest kind of sampling tubes that you should use. We suggest also volume of aliquots, number of aliquots, and these things, so that we kind of have this harmonized. And we are also working on an SOP that kind of puts together all the aspects that we just talked about, which is like which samples should be collected, how should they be processed, and how should they be stored. And we encourage everybody here listening to this talk to actually join this very interesting and nice work, and then finalizing our SOP that it can then be used for everybody in AGI. And that would be the right time point for the poll. Should I just read through then yeah, okay, and just read through it. So in serum versus plasma biomarkers, which statement about serum versus plasma is wrong? Plasma is the cell-free supernatant obtained by centrifugation of anticoagulated whole blood. Serum is plasma minus clotting factors. Serum plasma can always be used interchangeably in every biomarker assay. Special blood cards allow separation of plasma from whole blood without centrifugation. Prolonged storage of blood before processing can affect biomarker levels. So the question again is, which statement is wrong? So that's perfect. So actually, there was nobody wrong in the first and the fourth and fifth one of the options, and then there was like a few people thinking that that serum is not plasma minus blood clotting factors, but most of the people actually decide for the answer serum plasma can always be used interchangeably. That's not true. Now, I've given one example where this was especially not true. That is, for example, for tau biomarkers and phospho-tau biomarkers, which don't work well in serum, but work for whatever reason well in plasma, we actually don't know. Then I give over to Puneet.
Yeah, welcome everybody. So you've heard from the first speaker basically about what are biomarkers, different criteria, and why they are so important. And then you heard just now from David, who basically went very nicely through the importance of having standard operating procedures. So the question really is, why are we doing this initiative now? I mean, we've been having spinocerebellar ataxia and other ataxia for many years, but why now? I think I want to give examples of biomarkers in the context of the importance that we are now at the stage where we actually have drugs on the horizon, or biological on the horizon. And really the biomarkers are driven by the fact that we need to be ready for these clinical trials, which are just about to occur. And so I'm going to talk about the spinocerebellar ataxia, which is the autosomal dominant ataxia, just because as a group, they really form a substantial number of ataxias. When you start subparsing, some of the recessive ataxia can be quite rare. But Friedreich's may be an exception, but by and large, many of the autosomal recessive ataxia are pretty rare. On the other hand, we have these SCAs, and the SCAs, for many of you who already know, they are CAG repeat or trinucleotide repeat, and because of the coding region of the gene, it ends up creating polyglutamine repeats. And there are new ways by which we can basically either tackle the repeats at the very get-go at the level of DNA, which is CRISPR. Then there's ways by which you can target RNA using antisense oligonucleotides or siRNA. And all of these are based on the fact that these polyglutamine repeats, the proteins, they tend to be toxic, so the gain of toxic function. So if you could somehow tamp down this toxic function, perhaps preserving the normal function, whatever that might be, you might end up with a disease. A little bit similar to Huntington's disease, where clinical trials are based on these. And then finally, we are learning from the toxic proteins themselves have different ways by which they're causing toxicity, a little bit like spokes of a wheel, and each of these are being addressed. But regardless, we would like to know how we would, which patients to target and how we would follow. So in this case, it's also important to know that many of these diseases have been tackled very well in mouse models. So there's a little box on the side showing that the preclinical models show a lot of possibility and promise for these disorders. So, okay, so with regard to these clinical trials, the question is, can you engage the target? So the target here would be many of these proteins, which are called ataxin proteins, for example. Can you reduce them by using the RNAi methods or the ASO methods? We don't really need a biomarker for diagnosis because it's a genetic test, but we would like to have a biomarker that if you were to reduce the levels, you are basically hitting the target, and that means that the pharmacological agent is working. The other area we want to know is, can we get a measure for disease onset, severity, and progression? So this could be the protein itself, for example, the ataxin protein itself, or it could be some of the proteins which have already been mentioned, alluded to earlier by David, for example, neurofilament light chains and tau, for example, in different disorders. And I'll go a little bit more into detail with those. And then finally, it would also be important to learn from these knockdown experiments other insights into pathogenic mechanisms, which actually might come out as maybe not part of the clinical trial, but we might learn something from the clinical trials, and I put that idea there. And then finally, these clinical biomarkers, if you really remember the first speaker about, we might be able to find different layers of people who have got more or less of these biomarkers suggesting that they're early or late in the disease, which might be important for inclusion criteria and stratification for clinical trials. So you might want to have bin patients based on the same disease of severity and compare them as they progress, as opposed to somebody very more severe or very little with almost no symptoms at all. So that's where inclusion criteria and stratification come along. Okay, so biomarkers in the fluids, bodily fluids, are a little bit difficult to detect. And it's nice that technology has kept pace with our needs. For example, we now can detect low levels of proteins such as neurofilaments in the blood and the CSF, and remarkably the blood, because it basically has to be leaked out from the brain into CSF, into the blood, and that can be detected. And the way it can be detected is by basically using high-end ELISAs, if you will. So ELISA are basically antibody and protein interactions, which are then associated with the fluorescence signal. But there's one technology called single molecule-based analysis, or SIMOA technique. It's commercially available by several different companies now. And what's good about this technique is that this antibody-antigen reaction is occurring in little small bins of which can or wells in femtomolar size, and that elicits a signal, and that signal is either yes or no. So almost like a Boolean logic, you can computerize that signal, and you can basically get a very good quantification, which is very important when you want to stratify or follow biomarkers such as neurofilaments or tau and things like that. So that's just a slide to give you the impression. Of course, there's a lot to learn, but this is sort of an introductory course on biomarker. But I just want to tell you about that. And the second technology is a Meso-scale Discovery. So this works a little bit different, but it's an electrochemiluminescence based method, where this electrochemiluminescence basically amplifies the signal, so small amounts of analytes or proteins can be detected again by this commercially available array plate. And the good thing about arrays is that you can also test multiple analytes using the same sample, which is very important when you have small amounts of patient sample material to go along with different groups all interested in their own biomarkers. So you can give each group their own, and each can test different analytes, and that little bit of sample can go a long way. So there are clearly other techniques on the horizon, but I just want to mention these two because they come up all the time in some of the papers you might read. Now, here's an example of a spinocerebellar ataxia type 3. This is one of the most common spinocerebellar ataxias. It's just an example, but similar stuff is experiments are being done for the other SCAs. And this is by a group, Len Petruccelli's group in Florida, where they basically have found a way. This is actually an MSD way in this particular slide, where they can detect ataxin 3. This would be a target. If let's say there were an SCA3 target, this would be a way to detect SCA3 in the spinal fluid and also in the plasma. Now, the amount in the plasma is higher, and probably not all brain derived, but the point here is just to show that there is an assay to look at ataxin 3. It would be nice, for example, if in an ASO trial, I think there's one on the horizon for by the company Biogen, and they'll probably want to see, for example, the spinal fluid ataxin 3 is hit and reduced, because that would mean that the target SCA3 by the ASO to SCA3 is really hitting the target. So that would be one example of a target. Another example, now not of a target actually, but now of a disease progression biomarker, is the neurofilament light chain, which David mentioned a couple of times. And that is, it's turning out to be a very promising biomarker, not just in ataxia, but also in a whole variety of neurodegenerative syndromes. It's a very predominant intermediate or cytoskeletal protein in neurons. And because it's so abundant and it's very stable, it can be basically followed. And as I told you, it can even be detected in blood because it's so highly abundant and still leaks out from the spinal fluid into the blood, or from the brain into the blood. So in this study, which is done by Wilkie et al., again, SCA3, just to keep the theme of SCA3, you can see that you can detect the neurofilaments. Not only do you detect it late in the ataxic period, but you also detect in the pre-ataxic period. So you might be able to see disease conversion, for example, with the neurofilament light chain. And this is just showing how the neurofilament light chain goes up as many years of the disease. And also it goes up based on the CAG repeat count, which basically means that it's probably going up along with the disease severity and age as well. But I have to tell you that these are still early days, and I have to forward it, but next slide. Yeah, so these are still early days, and we are finding that there is a confounding effect of age. So what I mean by that is, even normally, the neurofilaments, they do go up as a function of age. And so when you are having a disease, which is also we're following over time, we have to sort of remove the factor of the aging process on the neurofilament level and the disease process. And these are still studies in progress, because here's a study which is done for totally different reasons, just studying neurofilaments as a function of age. And you can see that there's a small increase on age when people are in the middle age, like 40 to 50, then slowly accelerates in the fifth or sixth decade of life. And then there's a more rapid acceleration after the age of 60, and then the slope is pretty much constant. So you can imagine somebody at a younger age with a more rapid disease, when they are being compared to an older patient, that neurofilament level might have to be sort of normalized by some sort of a nomogram. But these are still areas of active research, and another reason why biomarkers have to be really studied very carefully, both in control and patient populations over time. So I mentioned SCA3, and I think similar studies are going to come out for all the SCAs. And the question is, why can't we just pool all these SCAs because they're rare disorders? Why can't we just have them all together, increase our sample size, have a better power? But it's not so simple, because many of these SCAs, they progress at different rates. For example, SCA1 is the fastest, SCA2 and SCA3 are intermediate, and SCA6 is the slowest. So you can imagine something like neurofilaments might show a more progression in SCA1 and less in, for example, SCA6. Or that might be, an a priori assumption, but needs to be tested. But it would be very important to keep these separate. And that's where the biomarkers might also follow different trajectories, even beyond the disease trajectories. It's also ambitious to study all the ataxia without identifying funding mechanism in place. So it's likely that clinical trials will focus on each ataxia individually, just for practical reasons. People from different groups study different ataxias just based on their historic track records, and it's more likely that based on their track record, they can get funding on that particular ataxia. For example, I'm interested in SCA1, for example, so I might prioritize that. But there are clearly going to be different groups studying different ataxias, and it's important that we all come together at the end or compare results. But I think that's how the disease will we will learn more about these SCAs, and also the more rarer autosomal recessive ataxias as we build patient populations in those cohorts as well. Next slide. So here's where the patient cohorts come in, and where you all come in as young investigators if you're interested. So we have basically, I put them in two layers. The top layer is the American or the US or cohorts with some international flavor, and then below are the more of the European dominated cohorts. So in the US, we have the National Ataxia Foundation, which is basically funding by philanthropy some of the natural history studies, which are following patients clinically with clinical trials rating scales. And now added onto that are basically collection of biomarkers, whether fluid or imaging. I've of course focused on fluid, but imaging biomarkers are also being studied. And then there's ReadyScar and a University of Michigan study, which are basically looking at different common ataxias. ReadyScar is looking at SCA1 and SCA3 a little bit more in detail because we're taking people at risk. That's why it's called ReadyScar. And in University of Michigan, it's just on SCA3. And some of these studies you can see on clinicaltrials.gov as they come about, but just a little bit more detail on some of these additional SCAs. And these are studied funded by the National Institute of Health. So it's not philanthropy, but it's government. In Europe, there are similar studies. There's a Euroscar, which is, I would say, a sort of counterpart to the abroad looking at different SCAs. RISCAR, which is looking at risk, so I kept it below the ReadyScar, a little bit similar. And ESME, which is looking again at SCA3. So there's some sort of comparisons where you can make between the two, which is really good because you can imagine that we might have cohorts in the North America as a diagnostic, and then for verification we can use the European cohorts or vice versa. I don't think we've really done that at a global level, but that's what I would hope would happen with the SCA Global and the ARCA Global as we move forward, really using the cohorts in a very judicious manner. So this is what it looks like on a global scale. When I say global, it's good to see a map. And here I'm just showing a map on the US to show how we are basically the National Ataxia Foundation has really tried to go to spread it out so that patients are not too far away. I mean, US is still a pretty big country, and there are some places in the Midwest, for example, which are away. But if you're on the East Coast or the coast and the upper Midwest, there are a lot of places where you can come. The red sites are the CRCSCA, the National Ataxia Foundation. The green are ReadyScar, and then there's some overlap where you can have both sites. Some of them are MR sites, and there was also a panel where there was some European sites. And here are the European sites. This is maybe a little bit out of date, but they are basically the EuroSCA and the RISCA centers, again showing you where you are in Europe and how patients have to travel. And as investigators, if you're close to some of these sites, how you could send samples or be associated with some of these sites. All right, so similar maps are in place for the ARCA registry. I don't want to leave that out because I mentioned the SCAs, but the autosomal recessive ataxia are also very important. And the hope would be that we end up with a large-scale registry for these as well. Next slide. So I think the point here, which we've tried to make with the three talks, is to give you a broad overview. So not a lot of depth, but I think you will get an overview of the importance of biomarkers, the need for really standardizing these operating procedures. So when you get these valuable samples, they really mean something. And then finally, how you can compare these ataxias basically with a view for the clinical trial readiness, which is coming in the next few years. So I think it's very timely. And we would really encourage you all, as young investigators, to either contact us or to be associated with this. The fact you've attended means that you are interested. So keep your interest alive and contact us so that you can be part of this study as we move on with our working groups. So this is a good time to remind you that we are really keen to take question and answers. So you can place them in the Q&A or in preferably the Q&A, so they're all in one place. But I see there's some, if they're in the chat, we will also look at those too. But the Q&A would be a good point. And I think we will end there.
Well, thank you very much. At the moment, I don't see any questions in the Q&A, so maybe you've answered them already all. I nevertheless would like to start with one question. You talked a little about this non-specific biomarkers like neurofilament light and more disease-specific biomarkers like the ATAXIA 3 example you showed. So what would be your feeling? Is there a trend to use more this non-specific markers since they probably tell more about the course of the disease? Or do you think finally we might end up in very detailed biomarkers that are specific for each and every entity? What do you think? Where will we go?
Okay, I can start with this and then maybe David can follow. So I think that there are going to be some advantage to take biomarkers which are relatively non-specific like neurofilaments, just because we know so much about them. They are commercially available assays which are really proving to be very sensitive and precise. And as long as we have sort of made sure that the diagnosis is correct and they're being used in a clinical trial, there's not really much to worry about. Let's say that they have Alzheimer's or Parkinson's or something else which could also raise neurofilament levels. So I think that from that perspective, I think neurofilaments would be one, maybe tau or other proteins which are seen in other neurodegeneration would be others. But I do think that there is a value to identifying more biomarkers in a very disease-specific manner. This is where some discovery approaches will have to be used. The challenge, of course, is it's easier to do discovery for common diseases like Alzheimer's, where you have large cohorts and you can power discovery, which also requires a certain amount of power. So, but I think that that will be the eventual goal, and we'll have better techniques. And the discovery could be over multiple platforms, not just protein, but also RNA and metabolites. So that's where I think we will. And who knows, we might have a panel, not just one for disease onset and one for progression, or a panel to follow all. So maybe that would be another approach. But I'm curious to know what David and Filippo have to add to this.
Yeah, I actually agree. I think a panel is definitely something to go for. One of the reasons is also that if you look at the studies and you also sometimes see the variation of biomarkers, and they can also be sometimes, have a level that looks like the patient is diseased, but he may be not. So I think if you have a panel and you have more than one biomarker, you have a better chance for achieving better specificity in diagnosing something, but obviously, for example, for some contexts of use of biomarkers, let's say for target engagement, having a disease-specific biomarker is necessary. It would be definitely preferable over like NFL, which is not so, which would not be a target engagement biomarker. So it really it depends. And we also see in some diseases, for example, Alzheimer's disease, that NFL is a pretty late marker, and that some markers are much rising way before that, like A beta in the CSF or p-tau in blood. And I think so. Then we will see similar things in ataxias. That way, I think the panel is the way to go for sure. Yeah.
I perfectly agree with you, David and Puneet. And I want also to stress that as we learn more from neurodegenerative conditions, we also learn that proteins involved in neurodegeneration are also critical in neurodevelopment. So we have to find also some biomarkers for early stages of alteration neurodevelopment. And for example, neuroinflammation is very critical. So it happens, perhaps we will end up with biomarkers that are different in different disease stages. And the most important is that we can even integrate different biomarkers, biological markers with imaging biomarkers, wearable sources of biomarkers. And I want to personally encourage young investigators who want to work on disease models and animal models also to consider the useful power of their model and taking biomarkers also from the mice and also from the zebrafish, because this is another high value that we can, for example, compare biomarkers between different transspecies biomarkers. That would be very interesting.
Thank you. And now we have a question in the Q&A section from Lucy Stovikova. Thank you for your presentation and overview, please. What are the best biomarkers to analyze in Friedreich's ataxia? And then as another, I would like to do MRI analysis in Friedreich patients. Who is the best contact person? I think that's not about biomarkers, but the first part. What do you think is the best fluid biomarker for Friedreich ataxia?
For ataxia. Yeah, it's like Puneet showed for SEA3, the possibility to measure the protein involved in the conditions. It would be excellent. We have this chance for Friedreich ataxia, for ataxin, but there are other surrogate biomarkers coming out also from different disease stages. And I don't know about the MRI. There are several cohort consortium around the world. One recent consortium was publishing recently some state-of-the-art of the imaging status of patients with Friedreich ataxia, and the consortium is led by Ian Harding, who's also a leader in the working group for imaging here at AGI. So perhaps the best person to get in touch with is Ian Harding.
I would add to that in Friedreich's, it's a loss of function of the protein frataxin 1. So most of the therapy they're trying to increase the levels. The question is, can we see frataxin going up? And there are quite a few drugs in the pipeline there. And I think in the US, we have a study being done by UPenn, David Lynch's side. I think he's probably the person to contact for Friedreich's, including imaging biomarkers.
And maybe for the European sites, this would be Katrin Reetz and the team in RWTH Aachen in Germany.
Okay, so there's another question about good biomarkers for SCA1.
There's a paper from Carlo Wilkie and me actually in neurology, which you might look into. This so NFL is a good. It seems to be a good biomarker there, also for preclinical forms of it.
And Puneet, any updates about ATAXIN 1 assays?
Yeah, so we actually in our own lab, we have an assay which is aimed similar to the MSD assay, but it's a low abundant protein, so that would be one. But I do know that the neurofilaments are definitely there. You know, we actually use a lot of mouse models, and neurofilaments definitely are panning out even in mouse models as well. So agree.
Okay, then I think we are happy to thank you for this very interesting talk. I learned a lot, and I think you perfectly got the scope. So to also give very basic information that are needed and helpful. So thanks a lot for this wonderful presentation.
Thank you very much for
Very
your.
much.
Bye.
Take care.
And bye and see you.